This demonstration is how to use the PhysioEx simulations to generate or to write a lab report. So students will be responsible for completing assessing starch digestion by salivary amylase, assessing pepsin digestion of protein, assessing lipase digestion of fat. For this demonstration, I am going to use amylase substrate specificity, okay, as a demonstration. So the link, you will click on the link, it will take you to the PhysioEx pertaining to that particular simulation. There you'll have the objectives, and these objectives are what you as a student will be expected to learn. You'll click next. You have an introduction telling you all about substrates and enzymes and all this other good stuff. So read up on the introduction. Then you have, it will tell you the material and methods that you're going to use. In your lab report, you are not going to list these equipment and reagents. You are not going to list this. When you're writing your material and methods, you have to write complete paragraphs on each of these different tests that you're doing. Let me show you an example. So here is an example. Okay, when you're writing your material and methods, do not include a list of materials that you use. These will be incorporated in your paragraphs. Here is a paragraph, and this paragraph is describing what I'm about to do. So to examine the effect of amylase substrate specificity, 6 tubes were prepared with different combinations of amylase, cellulase, peptidase, bacteria, and so on. So there I am saying what is going to be included or what were included in each test tube, but I am not listing these things. So once you write your material and methods in paragraph form, a reader will be able to understand and select and pick out what you actually use during the experiment. Of course, this is a simulation, but it pertains to the very same thing. So this is one way to do it. Here is another way you could do it. Okay, let me make this a little bit larger. Okay, so we have one way to examine the effect of amylase substrate. Here is another way you could write it. To examine the effects of amylase substrate specificity, 6 tubes were prepared with different combination of enzyme substrates, buffer, and water. Amylase, which breaks down starch, was added to tubes 1 to 3. Starch was added to tubes 1 and 5, and so on and so forth. But I'm not listing any of these things in the list I showed you earlier on. So going back to the simulation. So this is what you will be using in the simulation, but in your report, paragraph form. There will be questions, so you will answer these questions, try to answer them correctly. You read the introduction, try to understand them correctly. You will select one, check your answer, and it will tell you if it is incorrect, if it is correct. Next, you go to the next. Okay, so there are questions. Click one randomly, check, didn't come up, but it's going to be recorded. Cellulose and starch, let's see if we can get this one correct. Cellulose and starch are found in cell wall of plants, polymers of glucose. They are polysaccharides. They are polymers of glucose found in cell wall of plants, polymers of glucose, and polysaccharides. So let's try this one and see. So this is correct. Okay, so it tells you if it's correct, it tells you if it is incorrect. I think if you try to go to the previous one, you may not be able to correct something that you have incorrectly. Okay, more questions, and randomly I'm just picking one, continuing, another question, continuing, and then it will ask you to submit, so you submit. Alright, submit means these things are going to be recorded. Now for the simulation. So it says incubation, drag a test tube to the first holder in the incubation unit. Okay, and the 5 more will follow. So you drag this in the incubation unit, you just follow the instructions given. Okay, once you're finished with this, you go to next. So you always, once you finish with a task, you could go to next, then you follow the instructions. So this particular one has 6 tubes, and each of these must be added to each tube. So here you have a substrate, this is the enzyme, and of course you have buffer. In a lab, and normally you will add water or buffer first. So in this case, we're going to add the buffer first. Enzymes are the last thing you add when you're adding several different reagents to a reaction. So let's add pH 7 buffer. Now take track of the number of drops. All right, number of drops, because you need to include this in your material and methods. So it says add buffer 7, pH 7. So we go here, we add pH 7, 2, 3, 4, 5, 6. It's about 6 drops. You add to each tube. Okay, we could double check. 7, 7, 7, 7. Yeah, so we add to each tube. Add to each tube. Okay, next, starch. So we're going to add the substrate. So the first one is starch. So we add starch. 1, 2, 3, 4, 5, 6. It's about 6 drops, I think. Tube 2 is glucose. Tube 3 is cellulose. Tube 4 is cellulose. Tube 5 is starch. And tube 6 is cellulose. Okay, now for the enzyme. Tube 1 is amylase. Now you could add amylase. We know amylase is in 1, 2, and 3. So we're going to add amylase to the first 3. 1, 2, first 3, amylase. Then afterwards we add, this one is deionized water. Of course it's not an enzyme, but this is like a negative control. So we have deionized water in tube 4. In tube 5 we have peptidase, which breaks down peptides. And finally tube 6 we add bacteria. Once you finish adding, you don't want to do undo or reset unless if you have made an error. You go to next. There's a question, you answer the question, you check your answer, and then you go to next, submit your question, you go to next. Okay, select incubate to start the run. So we go to incubate. This is the timer. It says about 1 hour, 60 minutes. Of course, this is not really 60 minutes, but okay, so now it's done. Okay, so it takes about 10 seconds, but in real time it would have been 60 minutes. You go to next. Do you think test tube 3 will show positive Benedict's result? So it doesn't matter if this is test tube 3. Just pick one randomly, submit. And now it says drag the test tube in the incubation unit to the first small assay tube. Follow the instructions, you take this to the first small assay, and all the others will follow. So this is just a matter of following instructions. Tells you to do this, you do this. Tells you to do that, you do that. Okay, so now that is done, we go to next. Drag the iodine, okay, and again count the number of drops, add iodine. Good. And once you're finished, go to next. Record the data. Next. Once you finish, you go to next. Questions, you check your answer and you continue, submit, and you go to next. Well, here is where there was an error, so we can always go to the previous. Okay, where it says drag Benedict's reagent dropper, so we add Benedict's to this, count the number of drops. Next. says select boil, so you're going to boil the reaction. It doesn't tell you how long, but in the lab generally we do this for about 3 minutes. Okay, next it will tell you to record, so let's go to next. It says record the data. Inspect, greenish, it gives you information, and then it says record the data. Okay, so this is really similar to what you would do in a lab for that particular experiment. Now there's a question, so it asks you question, you check your answer, you say okay, you submit. Congratulations, you have completed this experiment, so select submit. Okay, submit. There are post-lab questions. It's going to ask you post-lab questions, so here you go. You answer the question. All right, you can check the experiment and you go to next. Again, you could check the experiment to answer the question. Okay, just randomly pick, go to next, check your thing, click on the one you think, click next. Same thing here, see this, click on what it's asking you and you think is correct, next, and then you submit. Okay, there are other questions. Okay, so you answer these questions. Again, this is not the one you're supposed to do, but the others are very similar. Submit, and when you're finished, you go all the way down. It's going to tell you printable, savable version. You click on this and you write your name. All right, you write your name, and your name will appear on your report. When you're finished, you press print and save as PDF. It gives you the option to save to Google Drive, up to you. PDF, it will save as PDF in Google Drive anyways if you are logged in. You save it, and now you have completed that lab report, that particular report. Okay, now what do you do with this? This data, this table, you need to include in your complete lab report. All right.